Welcome dear students. In this topic we are going to discuss a very important metabolic pathway, that is the glycolysis. My dear students, the word glyco, it refers to glucose, and lysis means breakdown. So basically glycolysis is the breakdown of the glucose. And my dear students, when a glucose molecule, which is an aldose as well as a hexose having 6 carbon atoms and aldehyde functional group, when that is broken down through a series of reactions, then we get two molecules of pyruvate. Now pyruvate is a 3 carbon compound, so the two pyruvates will be having 6 carbons of the glucose. And during this breakdown, my dear students, two ATP molecules are used and four ATP molecules are produced. So there will be a net gain of the two ATP molecules. Similarly, during this process, 2 units of the nicotinamide adenine dinucleotide, that will be reduced, and we will get 2 units of the reduced nicotinamide adenine dinucleotide. So that is an overall reaction of the glycolysis. Now this reaction of the glycolysis, it takes place in the cytoplasm, and cytoplasm of both plant cells as well as the animal cells. And my dear students, it took 100 years for the biochemists to understand the process of glycolysis, and there were three biochemists that played a major role. One of them is the Embden, second one is the Meyerhoff, and third one is the Pernas. Because these three biochemists, they played a major role in understanding the phenomena of the glycolysis, so that's why glycolysis is also called as the EMP pathway, after the name of these three biochemists. So that was the introduction of the glycolysis. Now, my dear students, you can see that I have mentioned two ATP molecules are used in the process of glycolysis and four ATPs are produced. So the glycolysis basically takes place in the two phases. And in these two phases, there are total 10 reactions. And my dear students, the one phase in which the ATP molecules are used, or you can say energy is used, that is called as investment phase of glycolysis or preparatory phase of the glycolysis. And the second phase in which the 4 ATP will be produced, that will be called as the yield phase or oxidative phase of the glycolysis. And there are total 10 steps in this process. The 5 steps will be in investment phase in which the 2 ATPs will be used, and the 5 steps will be in yield phase in which the 4 ATPs they are produced. So overall you can say that there is net gain of the 2 ATP molecules. Now let us see how this process of the glycolysis takes place. If you are a student of the medical sciences, it is very important for you to understand this glycolysis in a very well manner, including the structure of all those molecules that are involved in this glycolysis. So instead of just cramming the names of the different reactants and products of the glycolysis, you must also memorize their structure. So let's start from glucose. This one is the alpha D glucopyranose ring. So this one is alpha D glucose. And my dear students, this carbon is the sixth carbon of the alpha D glucose. In the first step, phosphorylation of this glucose takes place, and for that phosphorylation, one ATP molecule is converted to one ADP molecule, and the phosphate group released here that will get bonded to the sixth carbon of the glucose, and you will get glucose 6 phosphate. So with this sixth carbon, this phosphate group gets attached, and phosphorylation of glucose takes place, and you will get glucose 6 phosphate. And the enzyme which will be involved in this reaction, that will be a hexokinase enzyme. And in the liver cells, the glucokinase is involved. Hexokinase and glucokinase are isomerases, basically. So, whenever the word kinase will be written in this mechanism of the glycolysis, in that step, either the phosphorylation takes place or dephosphorylation takes place. Either the phosphate group will be added or the phosphate group will be removed. Now, in this second step, which is a reversible step, this glucose 6 phosphate is isomerized to its ketoisomer, and that ketoisomer is basically fructose, and with this sixth carbon of fructose, again this phosphate group is attached, so that will be called as the fructose 6 phosphate. And now, my dear students, this reaction is isomerization reaction, and this reaction is catalyzed by the phosphoglucoisomerase. So, enzyme involved is the phosphoglucoisomerase, which has converted glucose 6 phosphate into the fructose 6 phosphate. My dear students, the third step and the most important step, because that is the rate determining step of this process of the glycolysis, it is again phosphorylation of this fructose 6 phosphate, and for its phosphorylation again an ATP that will be changed to an ADP, and the phosphate group released that will be joined with this carbon number 1 of fructose 6 phosphate, and it will be converted to fructose 1,6 biophosphate. So with this first carbon again a phosphate group will be attached, and now this one will be fructose 1,6 biophosphate. And my dear students, this is again the phosphorylation, and this reaction will be catalyzed by the fructose 6 phosphate kinase. And my dear students, now in the next step, this fructose 1,6 biophosphate that is broken down into two smaller sugars, both are trioses. First is the aldehyde. This one is the first carbon, second and third, because with the third carbon of this triose, the phosphate group is attached. So that is called as glyceraldehyde 3-phosphate, G3P. And second, its isomer is produced, which is basically a ketone. It is a ketose. And that ketose is called as the dihydroxyacetone phosphate. These are two trioses. So a hexose is converted to two trioses, which are functional group isomers of the each other. And my dear students, now this reaction that will be catalyzed by an enzyme which will be called as fructose 1,6 biphosphate aldolase. So its complete name of this enzyme will be fructose 1,6 biphosphate aldolase. Now, my dear students, this was the step 1 of this glycolysis. It was the second step. This one was the third step. This one is the fourth step. And in the fifth step, this dihydroxyacetone that will be changed to this G3P, because the glycolysis process next that will happen with G3P and dihydroxyacetone phosphate that is converted to the G3P. This is again an isomerization process. It is a reversible step, and this one is the fifth step of the glycolysis. And this fifth step is catalyzed by an enzyme which is called as the triose phosphate isomerase. So it is triose phosphate isomerase. My dear students, in these 5 steps, the first phase of the glycolysis gets completed. That is the investment phase, which involves 5 different steps. And in these 5 different steps, there are 2 ATP molecules that are used and they are converted to the ADP molecules. So that was the investment phase or the preparatory phase of this glycolysis. Now in the next phase, the glyceraldehyde 3-phosphate that will be again phosphorylated and it will be changed to a molecule which will be called as 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. Now, my dear students, this reaction again, it is a phosphorylation reaction. So it will be catalyzed by an enzyme G3P kinase. Mean the complete name of the enzyme will be glyceraldehyde 3-phosphate kinase, and that will catalyze this sixth step of the glycolysis. And the first step of the next phase of the glycolysis, which is the yield phase, in which the energy in the form of ATPs and reduced NADH that will be produced. My dear students, in the next step, that is basically again dephosphorylation of this 1,3-biphosphoglycerate. So one phosphate group from this first carbon that is removed, and this phosphate group that will convert an ADP molecule into an ATP molecule. And now you will get a molecule that will be called as 3-phosphoglycerate. This one is the 3-phosphoglycerate, and the enzyme which will catalyze this seventh step of the glycolysis that will be called as the phosphoglycerate kinase. It will be phosphoglycerate kinase. Now, my dear students, I have missed a point here. In this step where the G3P is phosphorylated, In this step, the inorganic phosphate is basically added into this C3P molecule, and the inorganic phosphates, they are abundantly present in the cytoplasm where this glycolysis is taking place. And again in this sixth step, basically NAD is reduced to NADH. And my dear students, this hydrogen that is removed from the G3P, so you can say that this step is basically the dehydrogenation step, or you can say this step is the uh, oxidation of the G3P along with the phosphorylation. Now, my dear students, this 3-phosphoglycerate that is now changed to its isomer, that is 2-phosphoglycerate. So the 3-phosphoglycerate is isomerized to 2-phosphoglycerate. Again, it is an isomerization step, so that will be a reversible process. And this reaction is catalyzed by the phosphoglycerate mutase enzyme. And this is simply the mutation in which the position of the phosphate group is changed from third carbon to the second carbon. That is the eighth step of this glycolysis process. Now, in the next step, the 2-phosphoglycerate that is changed to this molecule, which is called as phosphoenol pyruvate. And my dear students, you can see that this hydrogen and this OH, they have joined to form water molecule, and this is a dehydration step in which the 2-phosphoglycerate is dehydrated to the phosphoenol pyruvate. And my dear students, this ninth step is catalyzed by an enzyme which is called as the enolase. And this is a dehydration reaction. That was the ninth step of the glycolysis. Now, in the final step, my dear students, you will get a molecule that is pyruvate. And again, the dephosphorylation of this phosphoenol pyruvate takes place, and the phosphate group which is released, it will convert an ADP into an ATP. And you will finally get a molecule that will be the pyruvate, and it is the final product of the process of the glycolysis. And this final step is catalyzed by pyruvate kinase. So this one is the 10th step of this process of glycolysis. Now, my dear students, starting from this sixth step, the yield phase starts. In the sixth step, 7, 8, 9, and 10, these are the 5 reactions of this second phase of the glycolysis. And you can see that when one molecule of the glyceraldehyde 3-phosphate is changed to one pyruvate, then 2 ATP molecules are produced. And when the second molecule of the glyceraldehyde 3-phosphate that will be again converted to pyruvate, 2 more ATP molecules will be produced. So there will be total production of the 4 ATP molecules in this yield phase of the glycolysis. And the 2 ATPs are used in the first phase, 4 ATPs are produced in the second phase. The net gain of the ATP, that will be 2. Similarly, my dear students, you can also see that when one G3P is converted to pyruvate, one reduced NADH is produced. And when the second G3P will be converted to pyruvate, then you will get another molecule of the NADH. So in this way, 2 NADH molecules and 2 ATPs, they are the products of the glycolysis, but the main process is the conversion of one glucose molecule into the two pyruvate units. So that was all about the glycolysis. I hope so that this lecture will be very understandable for you people. In case you find any problem, do not hesitate to ask. I will answer. Till then, take care.